Everyone is different when it comes to seeing improvements on antidepressants, but people typically start noticing positive changes after about 4 to 6 weeks of treatment, and it can take several months to feel the full effect of the medication. This graph shows you the onset of action of antidepressants in terms of its synaptic effects, its side effects, as well as the therapeutic effects. So the monoamine hypothesis stated that depression is due to reduced availability of monoamines, especially serotonin and norepinephrine. So antidepressants should be able to exert their therapeutic action almost immediately, corresponding with the increasing availability of monoamines at synaptic level. But that's not the case, as therapeutic effect develops only after several weeks. So, as I've mentioned earlier, antidepressant effects are generally not seen until 2 to 4 weeks of continuous treatment. So the question is, how can we explain this discrepancy between the immediate synaptic effect of antidepressant, in which the neurotransmitter levels are increased, and the delayed therapeutic or clinical effects? And in the next few minutes, we will explore the hypotheses that might explain the delayed therapeutic effect of antidepressants. The current prevailing hypothesis on why antidepressants take so long to work is because of the neuroadaptive changes that might happen in postsynaptic receptor signaling after the subsequent increased levels of serotonin or norepinephrine in the synaptic cleft, as well as the potential compensatory changes in gene transcription and the consequent structural changes in various brain regions and neurogenesis. We're first going to look at the neuroadaptive changes in postsynaptic receptor signaling. Over the years, researchers have also came up with the receptor hypothesis of depression. Basically, they don't think that the deficiency of monoamine neurotransmitter is the only reason for depression, but they came up with the receptor hypothesis of depression, in which they postulate that there are disturbances that occur in the function of receptors for the monoamine neurotransmitters. And such wrong receptor function may be caused by depletion of monoamine neurotransmitters, by abnormalities in the receptors, or by problems with signal transduction on post-receptor level. So it was then proposed that slow adaptive changes in post-receptor signaling and downstream mechanism could be possible explanation of delayed action of antidepressants. Also, they think it's most likely that post-receptor intracellular signaling cascade has got to do with changes in gene expression, neurotrophic mechanisms, and neurogenesis. To understand what I mean, let us take a look at the neurotrophic stress hypothesis of depression. A combination of stress, genetics, and environmental factors result in increased activation of the hypothalamic pituitary axis and chronic elevations in cortisol. The chronic elevation of cortisol downregulate the expression of multiple glucocorticoid-sensitive genes, including BDNF, or brain-derived neurotrophic factor. Reductions in BDNF result in neuronal atrophy and decreased synaptic density as seen here on this slide. Let's now take a look at what happens with the administration of antidepressants. Basically, in panel A over here is a timeline of what happens, or rather a timeline of events involved in mediating a therapeutic response to antidepressant therapy. So antidepressants or post-antidepressant administration, you would see a rapid increase in synaptic levels of monoamines, which happens over a few hours. And over several weeks, an upregulation in the expression of BDNF would produce an increased growth of neurons and upregulation of biochemical pathways involved in synaptic transmission, resulting in reversal of depressive symptoms. Let's take a closer look at panel B on the bottom left of the slide. So antidepressants such as SSRI, SNRI, NDRI, TCA, or monoamine oxidase inhibitor produce a rapid increase in synaptic levels of serotonin and norepinephrine, which then stimulates cyclic AMP response element binding protein, or CREB, through different converging signal transduction pathways. CREB stimulation then upregulates the expression of brain-derived neurotrophic factor, which causes increased neuronal growth and plasticity as illustrated in this particular figure over here. To recap, the neurotrophic hypothesis of depression proposes that depression is associated with reduced brain levels of BDNF, or brain-derived neurotrophic factor, in the hippocampus. So BDNF, or brain-derived neurotrophic factor, is actually responsible to promote survival of nerve cells by playing a role in the growth, maturation, as well as maintenance of this cell. So the delayed clinical efficacy is due to the fact that the process takes effect further downstream of receptor activation. So it's not just as easy as increasing the synaptic level of monoamine neurotransmitter. It's also what happens after the monoamine neurotransmitters activate the postsynaptic receptors. So there are intracellular changes that happen, which basically means there are gene transcription changes and whatnots. And this is why a longer time is needed to actually show a positive response after the administration of antidepressants. Again, this is just a representation of what happens, or rather the representation of neurotrophic effects of antidepressants, and this is what is shown on the picture. On the left, you see the hippocampal pyramidal neuron in normal state, with innervations with monoaminergic, glutaminergic neurons and others as indicated. So the activity or plasticity of the neuron is regulated, of course, by the brain-derived neurotrophic factor and by transcription factor CREB. So you have BDNF and CREB. In the middle image are changes in the neurons after heavy stress or as a result of increased glucocorticoid levels. So the dendritic tree is reduced and the densities of synaptic spines is decreased as well. And probably this is in connection with the decreased expression of BDNF. And in the rightmost picture shows effect of long-term treatment with antidepressants, which induce opposite effects on dendritic tree and synaptic spine. So you actually see an increase in the dendritic tree as well as synaptic spine density, and this is probably due to the increase in the expression of neurotrophic factor BDNF. So these are the theories that are postulated to be the potential reason behind the delayed effect of antidepressants. Thank you very much for watching. If you find this video useful for your learning process, hit like.